So far, you have learnt about the Calvin cycle in C3 plants, where 3-PGA is the first product of carboxylation. Let's now study carbon fixation in plants that have the first product of the Calvin cycle as a 4-carbon compound. These plants are called C4 plants, and the process of carbon fixation is called the C4 pathway. The C4 pathway was discovered by M.D. Hatch and C.R. Slack in 1966. Hence, it is also called the Hatch and Slack pathway. Carbon fixation in C4 differs from C3 plants, as it has one extra step preceding the C3 Calvin cycle. C4 plants are found in hot and dry areas and are adapted to tolerate high temperatures and can respond to high light intensities. The transverse section of C4 leaves, such as maize, shows a specialized arrangement of cells around the vascular bundles called the bundle sheath on the inner side. They are closely arranged and are without intercellular spaces. These cells contain many chloroplasts and have thick walls made of suberin, which makes them impervious to gases. The outer side of the vascular bundles contains mesophyll cells with chloroplasts. The large number of chloroplasts in the bundle sheath cells surrounding the vascular bundles makes the vein appear dark green. Leaves whose anatomy shows vascular bundles encircled by bundle sheath cells are called leaves with Kranz anatomy. Kranz is derived from a German word which means halo or wreath. Examples of plants exhibiting Kranz anatomy are maize and sugarcane. In the C4 pathway, carbon dioxide is fixed in the mesophyll cells. The first step is the conversion of pyruvate to phosphoenol pyruvate by utilizing one molecule of ATP in the presence of the enzyme pyruvate phosphate dikinase. Phosphoenol pyruvate, a 3-carbon compound, accepts carbon dioxide to form oxaloacetate, a 4-carbon compound. This reaction is catalyzed by phosphoenol pyruvate carboxylase. This enzyme ensures carboxylation even at low concentrations of carbon dioxide because of its greater affinity towards carbon dioxide compared to rubisco. Rubisco is absent in the mesophyll cells of C4 plants. Oxaloacetate is quickly converted to either malate or aspartate and is transported to the bundle sheath cells through plasmodesmata, where it is decarboxylated back to pyruvate. Decarboxylation results in the release of a carbon dioxide molecule. Pyruvate is transported back to the mesophyll cells through plasmodesmata and is phosphorylated to phosphoenol pyruvate. Then the cycle of carboxylation is reinitiated. The released carbon dioxide is fixed in bundle sheath cells, which are rich in rubisco, through the Calvin or C3 cycle. Thus we find that C4 plants undergo carboxylation twice, that is, in mesophyll through the C4 cycle and in bundle sheath cells via the Calvin cycle. In C4 plants, the C4 and C3 pathways are separated in space as they occur in different cells, that is, mesophyll cells and bundle sheath cells. As per energy requirements, the C4 pathway is a little expensive compared to the C3 pathway. This is because the C4 pathway consumes 2 ATP molecules for every carbon dioxide molecule fixed, Therefore, fixation of 6 molecules of CO2 requires 12 ATP molecules. When we include the ATPs used in the Calvin cycle to the total ATPs used, the count goes up to 30, which are 12 more than in the C3 pathway. Though the C4 pathway is quite expensive, it is an efficient method of carbon fixation for plants living in hot and dry conditions.